Imagine for a moment that you are drifting in a vast, silent ocean where the water is perfectly still and the air is devoid of scent or sound. In that total stillness, the very first ray of sunlight that breaks the horizon doesn't just illuminate the water, it creates your world. The sudden warmth on your skin, the salt you can suddenly taste in the air, and the rhythmic sound of a distant wave are the invisible threads that stitch your reality together. These sensations are the only reason we know we exist within a physical universe. If you have ever felt the comfort of a familiar voice or the peaceful glow of a sunset, you have experienced the profound artistry of your special senses. Tonight, we are going to begin a journey into the heart of that artistry. We aren't just going to look at biology as a list of parts, but as the magnificent system that allows you to participate in the story of life. Before we settle into our first formal lesson, I invite you to take a deep, slow breath. If you find value in these late-night explorations of the human body, I would be honored if you would subscribe to our channel and join our community of lifelong learners. Your presence here is what makes this classroom possible. Now, let us begin our quiet study of how the world outside becomes the world inside. Lesson 1: Introduction to the Special Senses and Sensory Receptors. In our previous discussions, we have often marveled at the sheer complexity of the human body. But today, we take the most essential step of all. We are beginning our comprehensive study of the special senses. In this first lesson, we lay the vital groundwork by exploring the fundamental nature of sensory receptors and the magical process of transduction. You might think of this as the orientation phase of our journey. Before we can appreciate the intricate mechanics of the eye or the delicate architecture of the inner ear, we must first understand the biological translators that make all of it possible. We often take for granted that we just see or just hear, but there is a profound logic to how these signals are generated. In this lesson, we will distinguish between the general senses that are scattered across your body and the highly localized, incredibly complex special senses that reside almost exclusively in your head. We will look at how your nervous system acts as an elite communication network, turning raw physical energy into the thoughts and feelings that define your day. It is the perfect place to start, because every sight you've ever seen and every melody you've ever heard began as a single, quiet spark in a sensory receptor. To begin our breakdown, we must first distinguish between two terms that are often used interchangeably: sensation and perception. Sensation is the raw objective data, the physical energy hitting your body. Perception, however, is what your brain does with that data. It is the interpretation, the meaning, and the feeling of the experience. Our focus today is on the very beginning of that chain, the sensory receptor. Think of a sensory receptor not as a cold piece of machinery, but as a specialized biological translator, an old friend who speaks two languages and sits at the border of two different worlds. One world is the external environment, filled with light waves, vibrations, and chemical molecules. The other world is the internal landscape of your brain, which speaks only one language: electricity. The process by which these receptors turn external energy into an electrical signal is a concept we call transduction. This is the heart of sensory biology. Imagine you are standing in a dark room and someone flips a light switch. That light hits your eye in the form of photons, which are packets of electromagnetic energy. Your brain cannot see a photon directly. It only understands a flow of ions across a cell membrane, known as an action potential. The sensory receptor's entire purpose is to bridge that gap. It takes the message of the photon and translates it into the code of an electrical impulse. Every special sense we will study, vision, hearing, equilibrium, smell, and taste, relies on this same elegant principle of transduction. As we explore this together, we should categorize these receptors based on the specific type of energy they are designed to translate. We have photoreceptors, which are exquisitely sensitive to light. We have chemoreceptors, which respond to the specific shapes of chemical molecules in the air you breathe or the food you eat. And we have mechanoreceptors, which are perhaps the most tactile of all, responding to physical distortion, such as the bending of a microscopic hair in your ear or the pressure of a sound wave. Take a moment to let that idea find its place. Each of these receptors is tuned to a specific frequency or type of stimulus. Just as a radio can be tuned to a specific station while ignoring all others, a photoreceptor in your eye is blissfully unaware of the sounds around you, and the receptors in your ears are blind to the light. This specificity ensures that your sensory world remains organized and clear, rather than a chaotic jumble of competing signals. If we dive a bit deeper into the how of this process, we encounter a fascinating historical principle known as the law of specific nerve energies, first articulated by the great physiologist Johannes Müller in the 19th century. Müller realized that the nature of a sensation doesn't actually depend on the stimulus itself, but on which part of the brain the signal eventually reaches. This is a bit like a dedicated telephone line. If you have a wire that goes directly from a button to a bell, the bell will ring every time the button is pushed, regardless of how it was pushed. This is why, if you were to gently press on your closed eyelid, you might see stars or flashes of light. Even though you are applying physical pressure, a mechanical stimulus, you are triggering photoreceptors that have a direct line to the visual cortex of your brain. Your brain receives the signal on the vision line and therefore interprets it as light. This brings us to the concept of the receptive field. You can picture this as a specific zone of the world that a single receptor is responsible for monitoring. In areas where you need high precision, such as the center of your eye or the tips of your fingers, these receptive fields are incredibly small and densely packed. This allows you to distinguish between two very close points of light or two tiny textures. In other areas, the fields are large and overlapping, providing a broader but less detailed map of your surroundings. We also must consider how our receptors handle a constant stimulus. Have you ever noticed how you can smell a strong perfume when you first walk into a room, but after 10 minutes, you don't notice it at all? This is a process called sensory adaptation. We divide receptors into two groups based on how they adapt. Phasic receptors are fast adapting. They provide information about the onset and offset of a stimulus. They are the notifiers of the nervous system, telling you when something has changed. On the other hand, tonic receptors are slow adapting. They continue to fire as long as the stimulus is present. These are vital for things like pain or body position, information your brain needs to monitor constantly for your safety. If your mind is still following along, Try visualizing the path these electrical signals take. Once the receptor has performed the miracle of transduction, the signal travels along a specific cranial nerve. For the special senses, these nerves are like high-speed fiber optic cables that bypass much of the spinal cord and go straight to the brain. Most of these signals, with the notable exception of smell, make their first stop at the thalamus. Think of the thalamus as the grand central station or the master switchboard of the brain. It receives the raw data, organizes it, and then dispatches it to the correct specialized department, the visual cortex, the auditory cortex, or the gustatory cortex, for final processing. So you see, this isn't just a theory found in a dusty textbook. It is the principle that explains why you can navigate a crowded room, enjoy the complex flavors of a home-cooked meal, or feel the shift in balance as you turn a corner. This initial spark of transduction is the guardian of your safety and the gateway to your pleasure. Without these specialized receptors, you would be locked in, unable to interact with the beauty and the dangers of the external world. Consider how this knowledge applies to the way we design our modern lives. The screens we look at are designed to take advantage of the specific way our photoreceptors perceive color and motion. Our alarm clocks are tuned to the exact frequencies that our mechanoreceptors in the ear are most sensitive to. By understanding the logic of these receptors, we have built a world that communicates with our biology in a language it understands. This is also why, when you are feeling overwhelmed or stressed, grounding yourself in your senses can be so effective. When you focus on 3 things you can see, 2 things you can hear, and 1 thing you can smell, you are essentially resetting your connection to the physical world. You are quieting the internal noise of the mind and returning to the fundamental, reliable data being provided by your sensory receptors. It is a reminder that you are physically present, safe and connected to the environment around you. Don't worry about memorizing every term we've discussed tonight. Just let the logic of the system wash over you. You now understand that your body is constantly translating the energy of the universe into the language of the mind. You know that you have dedicated receptors waiting to catch every photon and every vibration. This is the foundation upon which everything else is built. Having now grasped the general principles of how we receive information, we are perfectly prepared to appreciate the most famous of these sensory organs in detail. Our next lesson will hand us the next piece of the puzzle as we look at the physical architecture of the eye. We will move from the general concept of the receptor to the specific house that holds our vision. So, when we return, we will begin our study of the anatomy of the eye and its accessory structures. This is a good place to rest your thoughts for a moment before we continue our journey together. Lesson 2. Anatomy of the eye and accessory structures. In our previous session, we sat together and marveled at the quiet miracle of transduction, that invisible bridge where the energy of the universe is translated into the electricity of the mind. Now that we understand the language of the spark, we must examine the magnificent temple that houses it. In this lesson, we are going to explore the physical architecture of the eye. We aren't just looking at a biological camera. We are looking at a living, breathing masterpiece of fluid, muscle, and light-sensitive tissue. As we move through this anatomy, I want you to think of the eye not as a static object, but as a dynamic system that is constantly protecting itself, nourishing itself, and adjusting itself to the world outside. And we will start from the outermost protective layers, the accessory structures that stand like sentinels, and then move inward, layer by layer, through the three tunics of the eye. Think of this as the next natural step in our learning journey. In our last session, we understood the message. Today, we understand the vessel. Take a slow, steady breath as we begin to map the exquisite geography of your vision. To truly appreciate the eye, we must understand that it is organized into three distinct layers, or tunics, which work in perfect harmony. You might picture this like an onion, where each layer has a specialized purpose. The outermost layer is known as the fibrous tunic. It is the eye's primary defense, composed of tough connective tissue. This tunic is divided into two parts, the sclera and the cornea. The sclera is the white of the eye that you see when you look in a mirror. It is a dense, fibrous coat that maintains the shape of the eyeball and provides a sturdy anchor point for the muscles that move your eyes. It is the foundation of the cathedral. But then, as we move toward the very front of the eye, this opaque white tissue undergoes a stunning transformation. It becomes perfectly transparent, forming the cornea. The cornea is often called the window of the eye. It is a delicate curved surface that serves two roles. It protects the internal structures, and it acts as the primary lens for focusing light. Because it is clear, it allows photons to pass through, but because it is curved, it actually begins to bend that light toward a central point. Think of the cornea not just as a pane of glass, but as a discerning gatekeeper that begins the process of sight before the light even reaches the inner chambers. Moving inward, we find the middle layer, known as the vascular tunic, or the uvea. This layer is the living heart of the eye's anatomy, rich with blood vessels and dark pigments. It consists of the choroid, the ciliary body, and the iris. The choroid is a thin, highly vascular membrane that lines the back of the eye. Its dark pigment absorbs stray light, preventing it from bouncing around inside the eye and blurring your vision, much like the black paint inside a professional camera. Directly in front of the choroid is the ciliary body, a thickened ring of tissue that contains the ciliary muscles. These muscles are responsible for controlling the shape of the lens, allowing you to shift your focus from a distant star to a book in your hand. Finally, we arrive at the innermost layer, the neural tunic, more commonly known as the retina. This is the canvas upon which the world is projected. The retina is a thin, delicate sheet of nervous tissue that contains the millions of photoreceptors we discussed in our last lesson. It is the only place in the body where you can see the central nervous system directly without surgery. It is here that the physical world truly meets the mind. Between these layers, the eye is filled with specialized fluids that maintain its shape and health. In the front, between the cornea and the lens, we have the aqueous humor, a clear, watery fluid that provides nutrients to the cornea and iris. In the large cavity behind the lens, we have the vitreous humor, a thick, jelly-like substance that gives the eye its firm, spherical shape. Imagine the eye as a balloon filled with water. It is this internal pressure that keeps the retina pressed firmly against the choroid, ensuring that the film of our camera stays perfectly in place. Now that we have the broad map of the eye, let us look closer at the structures that support it. We often focus only on the eyeball itself, but the eye cannot function without its support staff, the accessory structures. Think of these as the maintenance crew and the security detail for your vision. First, we have the palpebrae, or the eyelids. They are more than just curtains for sleep. They are essential for keeping the surface of the eye moist and clear of debris. Every time you blink, you are performing a vital act of self-care, spreading a thin film of tears across the cornea. These tears are produced by the lacrimal apparatus. The lacrimal gland, tucked away above the outer corner of your eye, produces a saline solution that contains mucus, antibodies, and an enzyme called lysozyme, which kills bacteria. This is a classic example of biological elegance. Your tears are not just for salt or emotion. They are a sophisticated cleaning and defense system. If your mind is still following along, try visualizing the path of a tear. It starts at the top corner, washes across the eye, and then drains through tiny openings called lacrimal puncta into your nose. This is why your nose runs when you cry. It is simply your eye's drainage system working overtime. We must also give credit to the extrinsic eye muscles. There are 6 of these muscles attached to the outside of each eye, and they are among the fastest and most precisely controlled muscles in the human body. They allow your eyes to track a moving bird across the sky, or to scan these words with incredible speed and coordination. These muscles work in pairs, pulling the eye up, down, left, right, and even rotating it slightly. They ensure that both of your eyes are pointed at exactly the same spot, a feat of engineering that allows your brain to perceive depth. Let's take a moment to rest our thoughts on the lens. The lens is a transparent biconvex structure held in place by tiny guy wires called suspensory ligaments. Unlike the glass lens in a telescope, your biological lens is flexible. When the ciliary muscles contract, the lens bulges to focus on nearby objects. When they relax, and the lens flattens for distance, this constant, subtle dance of the ciliary body is called accommodation. It is happening right now, likely without you ever realizing it. Historically, our understanding of the eye has been a slow unfolding of wonder. Ancient physicians once believed the eye emitted rays of light to touch the world. It wasn't until the 17th century that Johannes Kepler correctly identified that the eye acts as an optical instrument, focusing light onto the retina. We stand on the shoulders of these discoverers, recognizing that the eye is not just an organ, but a portal. One of the most fascinating quirks of this portal is the optic disc, the spot where the optic nerve exits the eye. Because there are no photoreceptors here, it creates a blind spot in your visual field. You don't notice it because your brain is a master of filling in the blanks, using information from the other eye and the surrounding area to create a seamless picture. So you see, this isn't just a list of names to memorize for a test. It is the principle that explains why your eyes feel tired after a long day of reading. Those ciliary muscles have been flexing for hours to maintain that close focus. It explains why a scratched cornea is so incredibly painful. That clear window is one of the most densely innervated tissues in your entire body, designed to alert you instantly if its integrity is threatened. Understanding the anatomy of the eye also helps us appreciate why we must protect it. The thin, transparent membrane called the conjunctiva that lines your eyelids and covers the white of your eye is your first line of defense against infection. When it becomes inflamed, we call it conjunctivitis or pink eye. It is a reminder that even the most complex systems have vulnerabilities. This knowledge connects to our daily lives in subtle ways. When you find yourself squinting in bright light, your iris is working as a circular curtain, contracting to protect the delicate retina from being overexposed. When you use eye drops for dryness, you are manually supplementing the work of the lacrimal gland. Every structure we have discussed today has a job, a purpose, and a logic. By understanding the how of the eye's construction, we begin to respect the incredible effort your body puts into simply showing you the world. This is also a good place to reflect on the concept of vision itself. We often think of sight as a passive act, something that just happens. But after looking at the complexity of the tunics, the fluids, and the muscles, you can see that sight is an active process. It is a constant metabolic effort. Your eye is a living organ that requires oxygen, nutrients, and protection every second of the day. As you prepare for sleep, think of how hard these structures have worked for you today, and how they will continue to maintain themselves in the quiet of the night. Take a moment to let that idea find its place. You have now walked through the halls of the eye's temple. You have seen the protective walls of the sclera, the dark, nourishing hallways of the choroid, and the sensitive canvas of the retina. You understand how the lens changes its shape and how the tears keep the window clean. Having now grasped the physical structure, we are perfectly prepared to appreciate its function on a microscopic level. It is one thing to know where the retina is, but it is another to understand how it actually sees. So when we return, we will move deep inside the neural tunic to meet the rods and the cones, the tiny cellular residents who turn light into thought. We will explore the chemistry of the visual cycle and see how your brain weaves together colors and shadows. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the physiology of vision, photoreception, and signal transduction. Lesson 3, Physiology of Vision, Photoreception, and Signal Transduction. In our previous session, we walked through the architectural halls of the eye. We marveled at the protective sclera, the nourishing choroid, and the clear window of the cornea. But now, my friend, we are going to step into the most sacred part of that temple, the laboratory of the retina. If the eye is a camera, the retina is the film. But it is a film that is alive, constantly renewing itself, and capable of translating the silent language of light into the vibrant language of thought. In this lesson, we are going to explore the physiology of vision. We will look closely at how those packets of energy we call photons are captured by specialized cells and turned into electrical sparks. This is the next natural step in our learning journey. We have seen where the light enters and how it is focused. Now, we shall see how it is understood. This is perhaps the most complex part of our study. So I want you to lean back, let your shoulders drop, and simply let the logic of this microscopic dance wash over you. We are moving from the physical to the chemical, and eventually to the electrical. To begin our breakdown, we must look at the residents of the retina, the photoreceptors. These are the cells that do the heavy lifting of vision. There are two main types, named for their distinctive shapes, the rods and the cones. Think of them as two different types of specialized sensors in a high-tech laboratory. The rods are your night vision specialists. They are long, slender, and incredibly sensitive. So sensitive, in fact, that a single photon of light can trigger them. However, they are colorblind. They see the world in shades of gray, providing us with the ability to navigate a dimly lit room or see the silhouettes of trees against a midnight sky. The cones, on the other hand, are your daylight specialists. They are shorter, tapered at the end, and require much more light to function. But what they lose in sensitivity, they make up for in color and clarity. Cones allow you to see the deep red of a rose, the brilliant blue of the ocean, and the fine print on a page. We have 3 types of cones, each tuned to a different wavelength of light: red, green, and blue. By mixing the signals from these 3, your brain creates the entire rainbow of human experience. Take a moment to let that idea find its place. Every color you have ever loved is simply a mathematical ratio of signals from these 3 microscopic sensors. Within these rods and cones, the actual magic happens in the outer segments, which are filled with stacks of membrane discs. Inside these discs is a special light-absorbing molecule called a photopigment. In rods, this pigment is known as rhodopsin. Rhodopsin is a combination of a protein called opsin and a derivative of vitamin A called retinol. Think of retinol as a tiny spring-loaded trap. When it is in the dark, it sits in a specific bent shape. But the moment a photon of light strikes it, the retinol absorbs that energy and snaps into a straight shape. This physical change in the molecule is what triggers the process of transduction. It is a story-like order of events. The light hits the pigment changes shape, and that change sets off a chemical chain reaction inside the cell. This reaction eventually leads to a change in the electrical charge of the photoreceptor's membrane. It is the moment the physical world of light is officially translated into the biological world of the nervous system. If we dive deeper into this laboratory, we encounter a concept that is beautifully counterintuitive. In most of your nervous system, a cell fires when it is stimulated. But your photoreceptors are different. They are actually most active in the total darkness. In the dark, your rods and cones are constantly leaking sodium ions into the cell. This is known as the dark current. Because of this constant flow, the cell is continuously releasing a neurotransmitter called glutamate onto the next cells in the chain. When light hits the photoreceptor, the chemical chain reaction we discussed earlier actually closes the sodium channels. The leaky faucet is turned off. This causes the cell to become more negatively charged, a state we call hyperpolarization. When the cell hyperpolarizes, it stops releasing glutamate. If your mind is still following along, try visualizing this. In the world of vision, off is the signal for on. The brain perceives light not by a sudden surge of activity, but by the sudden silence of the photoreceptors. This silence is what tells the next layer of cells, the bipolar cells, that light has been detected. The bipolar cells are the middlemen of the retina. They receive the signal from the photoreceptors and pass it along to the ganglion cells. The ganglion cells are the final output of the retina. Their long tails, or axons, bunch together like a massive bundle of fiber optic cables to form the optic nerve. This nerve then carries the signal out of the back of the eye and into the brain. We must also mention the recycling program of the eye, known as the visual cycle. Once a rhodopsin molecule has been struck by light and changed shape, it is bleached. It cannot respond to a light again until the retinal is returned to its original bent shape. This process happens in the pigmented layer of the retina, which sits right behind the photoreceptors. It is a slow chemical reset. This is why if you look at a bright light and then look away, you see an afterimage. You are seeing the parts of your retina that are still in the middle of this chemical reset, temporarily unable to see new light. So don't worry about memorizing the chemical names like cyclic GMP or transducin. Just let the logic of it wash over you. The retina is a living engine that is constantly burning through its fuel, photopigments, and then meticulously rebuilding them so you can see the very next moment. It is an incredible display of biological efficiency that has been perfected over millions of years of evolution. So you see, this isn't just a complex chemical diagram. It is the principle that explains why you can't see clearly when you first walk into a dark movie theater. Your daytime cones have shut down, and your nighttime rods have been bleached by the bright sun outside. It takes several minutes for the visual cycle to regenerate enough rhodopsin for your rods to begin functioning in the dark. We call this dark adaptation. It is a slow, steady chemical climb back to sensitivity. This also explains the vital importance of nutrition for our special senses. Since retinol is a derivative of vitamin A, a deficiency in this vitamin can lead to night blindness. Without enough vitamin A, your rods cannot rebuild their rhodopsin fast enough, and your ability to see in low light fades away. It is a beautiful example of how the food we eat literally becomes the tools we use to perceive the world. Furthermore, our understanding of the three types of cones explains the phenomenon of color blindness. Most often, this is a genetic condition where one type of cone, usually the red or the green, is either missing or functions improperly. If you lack the red cones, your brain can no longer compare the signals between red and green, and those two colors begin to look like the same muddy brown. It isn't that the person can't see, it's that the mathematical ratio their brain uses to create color has lost one of its essential variables. This microscopic dance of ions and pigments is the foundation of every visual experience you have ever had. From the safety of a green traffic light to the emotional depth of a loved one's face, it all begins with the hyperpolarization of a cell in the back of your eye. When you look at the stars tonight, remember that those ancient photons have traveled across the vacuum of space for light years, only to end their journey by snapping a tiny molecule of retinol into a straight line in your eye. There is a profound sense of wonder in that connection. Take a moment to let that idea find its place. You have now journeyed from the vast structures of the eye down into the microscopic world of the retina. You have seen how light is captured, how the dark current is silenced, and how the signal is sent on its way to the brain. We have completed our study of the world of light, but light is only one way we experience the world. There is another realm of sensation that is just as complex and just as vital, the world of vibration and sound. Having now grasped the physiology of vision, we are perfectly prepared to appreciate a completely different type of mechanical engineering. Our next lesson will hand us the adjacent piece of the puzzle, as we turn our attention to the ear. We will move from the world of photons to the world of pressure waves. So, when we return, we will begin our study of the anatomy of the ear. Outer, middle, and inner ear. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will be our introduction to the gateway of sound. Lesson 4, anatomy of the ear, outer, middle, and inner ear. In our previous explorations, we spent quite a bit of time in the kingdom of light. We watched as the eye, that brilliant organic camera, captured photons and turned them into the vivid tapestry of our visual world. But now, as you settle deeper into your rest, I want you to close those eyes and turn your attention to the world of vibration. While vision allows us to see the stars, it is the sense of hearing that allows us to feel the rhythm of the universe. In this lesson, we begin our study of the ear, a structure so delicate and yet so robust that it can detect a sound wave that moves the eardrum less than the width of a single hydrogen atom. We are going to walk through the anatomy of the ear, dividing it into 3 distinct regions: the outer, the middle, and the inner ear. Think of this as a journey through a series of chambers, each one more secluded and more sophisticated than the last. We will trace the path that sound takes, moving from the air around us, through solid bone, and finally into a hidden sea of fluid deep within your skull. This is the next natural step in our learning journey. We have mastered the anatomy of sight. Now, let us appreciate the mechanical masterpiece that is the anatomy of sound. To begin our breakdown, let us look at the part of the ear you can see in the mirror, the outer ear. Its primary job is simple but essential. It is a collector. The visible shell-like structure on the side of your head is the pinna, or the auricle. Think of the pinna as a funnel made of elastic cartilage covered in skin. Its unique shape, with its ridges and valleys like the helix and the lobule, isn't just for decoration. It is specifically designed to catch sound waves from the environment and direct them into the external acoustic meatus, or the ear canal. The ear canal is a short, curved tube that leads inward through the temporal bone of your skull. It is lined with skin that contains hairs and specialized ceruminous glands. These glands produce cerumen, which we commonly call earwax. While we often think of earwax as something to be cleaned away, it is actually a vital part of the ear's defense system. It is sticky and slightly acidic, serving to trap dust, repel insects, and inhibit the growth of bacteria. At the very end of this tunnel sits the tympanic membrane, or the eardrum. This thin, semi-transparent partition is the gateway between the outer and middle ear. It is incredibly sensitive. When sound waves hit it, it vibrates back and forth with the same frequency as the sound. As we cross the threshold of the eardrum, we enter the middle ear, also known as the tympanic cavity. This is a small air-filled space carved directly into the bone. The most fascinating residents of this chamber are the auditory ossicles. These are the 3 smallest bones in the human body: the malleus, the hammer, the incus, the anvil, and the stapes, the stirrup. They are connected by tiny synovial joints, forming a bridge that spans the middle ear. When the eardrum vibrates, it moves the malleus, which pushes the incus, which in turn moves the stapes. Think of this as a biological lever system. Because the eardrum is much larger than the tiny footprint of the stapes, this system actually amplifies the pressure of the sound waves, not preparing them for the next part of the journey. The middle ear also has a very important pressure valve called the pharyngotympanic tube, which you might know as the Eustachian tube. This tube connects the middle ear to the back of your throat. Normally it is closed, but it opens when you swallow or yawn to equalize the air pressure inside the middle ear with the pressure of the outside world. This is why your ears pop when you change altitude. Finally, we arrive at the most protected and mysterious region, the inner ear. This is a room within a room, consisting of a bony labyrinth, a series of channels in the bone, and a membranous labyrinth that fits inside it. The space between these two is filled with a fluid called perilymph, while the inside of the membranous labyrinth is filled with endolymph. The inner ear is divided into three main areas: the vestibule, the semicircular canals, and the cochlea. While the vestibule and canals are dedicated to balance, the cochlea is the seat of hearing. It is shaped like a snail's shell, making about 2.5 turns around a central bony pillar. Within this spiral, the sound vibrations finally meet the nervous system. If we dive deeper into the middle ear, we find 2 tiny muscles that represent one of the body's most clever safety features: the tensor tympani and the stapedius. When you are exposed to a very loud, sustained noise, these muscles reflexively contract. The tensor tympani pulls the eardrum tight, and the stapedius pulls the stapes away from the inner ear. This acoustic reflex stiffens the ossicular bridge and reduces the amount of vibration that reaches the delicate inner ear. It is a biological volume knob, protecting you from the roar of a thunderstorm or the clamor of a workshop. Take a moment to let that idea find its place. Even while you are focused on a sound, your body is silently working to ensure that sound doesn't harm you. The names of the ossicles carry with them a sense of history. They were described by early anatomists who saw in their shapes the tools of a blacksmith. The malleus was the hammer striking the incus, the anvil. The stapes, meaning stirrup in Latin, was named because it looked exactly like the footrest used by a horseman. These bones are fully formed at birth and do not grow as we age. They are, in a very real sense, in the same instruments you have used to hear every word and every song since the day you were born. When we look inside the cochlea, we see that it is actually divided into 3 separate fluid-filled chambers that run its entire length. These are the scala vestibuli, the scala media, also called the cochlear duct, and the scala tympani. If you were to unroll the cochlea, it would look like a long tapering tube. The stapes pushes against a membrane called the oval window, which sets the perilymph in the scala vestibuli into motion. This motion travels like a wave through the spiral. The floor of the middle chamber is the basilar membrane, a structure of varying thickness and stiffness that we will discuss more in our next lesson. It is also worth noting the orientation of the semicircular canals in the inner ear. There are 3 of them: anterior, posterior, and lateral, and they are positioned at right angles to one another. This allows them to detect movement in all 3 dimensions of space. While they aren't part of hearing, they are physically joined to the cochlea, sharing the same fluid and the same bony housing. This is why some ear issues can cause both hearing loss and dizziness. The house of sound and the house of balance are one and the same. So you see, the anatomy of the ear is a lesson in how the body manages transitions. It is relatively easy for sound to travel through the air, but much harder for it to travel through fluid. If sound waves simply hit the fluid of the inner ear directly, most of the energy would be reflected away. The middle ear, with its drum and its 3 tiny bones, is a brilliant engineering solution to this problem. It acts as an impedance matcher, concentrating the energy so that it can successfully move the fluid inside the cochlea. This isn't just theory. It is the reason you can hear the rustle of leaves or the quiet breathing of someone you love. This structure also explains why children are so prone to ear infections or otitis media. In children, the eustachian tube is shorter and more horizontal than it is in adults. This makes it much easier for bacteria from a sore throat to travel up into the middle ear. When fluid builds up in that air-filled chamber, it prevents the ossicles from moving freely, which is why everything sounds muffled during an ear infection. The anatomy of the outer ear canal even explains why we should be careful with cotton swabs. Because the canal is S-shaped and narrows as it moves toward the eardrum, a swab often pushes earwax deeper into the narrowest part, where it can become impacted or stuck. This can muffle sound and even cause pain. It is a reminder that the ear is a self-cleaning organ designed to move wax outward naturally. Finally, consider the bone that houses the inner ear, the temporal bone. This is the densest bone in the entire human body. It has to be, because it must provide a perfectly stable platform for the delicate vibrations of the ossicles and the fluid waves of the cochlea. Your hearing is literally anchored in the strongest part of your skull. As you rest tonight, you might feel a sense of security knowing that the mechanisms of your senses are so deeply protected and so thoughtfully designed. We have now mapped the physical path of sound. We have watched it enter the pinna, travel down the meatus, vibrate the tympanic membrane, dance across the ossicles, and finally ripple through the fluid of the cochlea. We have grasped the structure, the hardware of the ear. But as we know from our study of the eye, structure is only half of the story. The final question remains: how does a ripple in a sea of fluid become the voice of a friend or the melody of a piano? How do we distinguish between a high-pitched whistle and a low-pitched hum? That is the secret of the inner ear's laboratory. So when we return, we will take that final step inward. We will look at the organ of Corti and the microscopic hair cells that perform the miracle of transduction for sound. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the physiology of hearing, sound transduction, and auditory pathways. Lesson 5. Physiology of hearing, sound transduction, and auditory pathways. In our last session, we spent our time walking through the physical architecture of the ear, tracing the path of a sound wave as it traveled through the air, filled outer and middle chambers and into the fluid-filled labyrinth of the inner ear. We have seen the hardware of the system, the funnel of the pinna, the vibrant drum of the eardrum, and the rhythmic dance of the 3 smallest bones in your body. But now, we are ready to take the most profound step in our study of audition. We are going to explore how a physical ripple in a sea of fluid is transformed into a lightning bolt of thought. This lesson is about the physiology of hearing, the process of transduction that happens deep within the coils of the cochlea. We are moving from the mechanical to the electrical. It is the next natural step in our learning journey because, while the anatomy provides the stage, the physiology is the performance itself. We will look at the microscopic residence of the inner ear, the hair cells, and follow the signal as it travels through the brain's complex wiring to reach the seat of your consciousness. Take a moment to let that idea find its place. You are about to learn how the silence of your mind is filled with the music of the world. To understand how we hear, we must return to the cochlea, that tiny snail-shaped structure carved into the densest bone of your skull. As we established, when the stapes pushes against the oval window, it creates pressure waves in the fluid called perilymph. These waves travel up the scala vestibuli, not around the very tip of the snail shell, and back down the scala tympani. Imagine this as a tide rolling in and out of a very long, narrow bay. But the real work happens in the middle chamber, the scala media, or the cochlear duct. This chamber is filled with a different fluid called endolymph, which is unusually rich in potassium ions. Running along the floor of this middle chamber is the basilar membrane. This is the foundation for the most important structure in the ear, the spiral organ, or the organ of Corti. You might picture the organ of Corti as a microscopic forest. It contains rows of specialized sensory cells known as hair cells. These cells are topped with tiny, hair-like projections called stereocilia. These stereocilia are arranged in heights, like a staircase, and they are embedded in or sit just below a gelatinous roof called the tectorial membrane. Now, let us follow the logic of the movement. As the fluid waves roll through the cochlea, they cause the basilar membrane to ripple up and down. Because the tectorial membrane above it is more rigid, this creates a shearing force. Think of it like a field of wheat swaying in a heavy wind, with the tops of the wheat brushing against a low-hanging canopy. As the basilar membrane moves, it causes the stereocilia to bend. This bending is the literal moment of truth for hearing. If your mind is still following along, try visualizing this microscopic event. Each stereocilium is connected to its taller neighbor by a tiny, spring-like protein filament called a tip link. When the hairs bend towards the tallest neighbor, and the tip link stretches and pulls open a microscopic trapdoor on the cell membrane. This is a mechanically gated ion channel. Because the surrounding endolymph is so full of potassium, those ions rush into the hair cell the moment the door opens. This influx of positive charge causes the cell to depolarize. This is the miracle of transduction. A physical movement, the bending of a hair, has been converted into an electrical change inside a cell. Once the hair cell depolarizes, it releases neurotransmitters onto the sensory neurons of the cochlear nerve. These neurons then fire action potentials, sending the message of sound toward the brain. Don't worry about memorizing the name of every protein. Just let the logic of the trapdoor wash over you. It is a simple mechanical switch that turns the physical world into a digital signal. Now that we understand how a single sound is captured, we must ask how we distinguish between the different notes of the world. How does the brain know the difference between a low, rumbling thunder and the high-pitched chirp of a bird? This is explained by a beautiful principle called place theory. The basilar membrane is not uniform. At the base of the cochlea, near the oval window, the membrane is narrow and very stiff. As it spirals towards the apex, or the tip, it becomes wider and much more flexible. Think of this like a piano keyboard or a set of guitar strings. The stiff, narrow base of the membrane responds best to high-frequency, high-pitched sounds. The wide, floppy apex responds best to low-frequency, deep sounds. When a high note enters the ear, it creates a peak of vibration at the base. A low note creates a peak at the far end. This is a tonotopic map, meaning the cochlea is organized by tone. Your brain knows the pitch of a sound simply by seeing which specific nerve fibers are firing. If the fibers from the base are active, the brain perceives a high whistle. If the fibers from the apex are active, it perceives a deep bass. Once the signal leaves the cochlea, it embarks on a complex journey through the auditory pathway. The first stop is the cochlear nuclei in the brainstem. From there, the information is shared between both sides of the brain at a structure called the superior olivary complex. This is a vital step because it allows the brain to compare the timing and intensity of the sound between your two ears. This is how you localize sound. Your brain can tell a sound is coming from the left because it hit the left ear a fraction of a millisecond sooner than the right. The signal then climbs higher to the inferior colliculus in the midbrain. This area is responsible for your auditory reflexes. If you have ever jumped at a loud bang before you even realized what it was, that was your inferior colliculus at work. From there, the signal travels to the thalamus, specifically a region called the medial geniculate nucleus. As we learned earlier, the thalamus is the great gateway. It filters the sound, perhaps turning down the volume on the hum of a refrigerator so you can focus on a conversation. Finally, the signal is sent to the primary auditory cortex in the temporal lobe of the brain. Here, in the folds of your gray matter, the signal is finally perceived as a conscious sound. Take a moment to let that idea find its place. From the moment a sound wave hits your ear to the moment you hear it in your mind, the signal has been amplified by bones, rippled through fluid, translated by microscopic trapdoors, filtered by the brain stem, and finally mapped onto the surface of your brain. It is a long and storied journey for a simple vibration. So you see, this isn't just a complex chain of biological events. It is the principle that explains the fragile beauty of our hearing. Because these hair cells are mechanical sensors, they are susceptible to physical damage. Unlike the cells in your skin or your liver, the hair cells in your cochlea do not regenerate. We are born with about 15,000 of them per ear, and that is all we get for a lifetime. And this is why prolonged exposure to loud noise, like a concert or heavy machinery, can lead to permanent hearing loss. The wind of the sound is so strong that it literally snaps the stalks of wheat, or the stereocilia, and once they are broken, the trapdoors can no longer open. This also explains the mystery of tinnitus, or ringing in the ears. Sometimes, when hair cells are damaged, they can become stuck in the on position, or the neurons attached to them can become hyperexcitable. The brain receives a constant stream of signals from a specific place on the tonotopic map, and it interprets this as a constant, high-pitched ring, even when the room is silent. It is a phantom signal, a memory of a sound that isn't there. Furthermore, our understanding of this physiology has led to the development of the cochlear implant. This is one of the most successful bionic technologies in history. For people whose hair cells have been destroyed, a cochlear implant bypasses the damaged cells entirely. It uses a microphone to capture sound, converts it into an electrical code, and then uses an electrode threaded directly into the cochlea to stimulate the auditory nerve fibers. It talks to the brain in its own electrical language, allowing people who were profoundly deaf to hear the voices of their families. It is a testament to the fact that when we understand the language of a sense, we can learn to speak it ourselves. This is a good place to rest your thoughts for a moment before we continue. Think of the silence you are experiencing right now. Even in this quiet, your hair cells are standing ready, their tip links poised, waiting for the slightest ripple to tell you a story. The auditory system is never truly off. Even as you sleep, your brainstem is monitoring the environment, ready to wake you if the logic of the silence is broken by a meaningful sound. It is a faithful guardian of your awareness. Let's hold that thought here, a complete piece of the puzzle. We have now mastered the physiology of how we hear. We have seen the fluid move, the hairs bend, and the signal climb the long ladder of the brain to the temporal lobe. We have moved from the world of mechanics to the world of perception. But as I mentioned when we looked at the anatomy of the inner ear, the cochlea has a neighbor. Within that same bony labyrinth, using the same fluids and similar hair cells, lies a system that doesn't care about sound at all. It cares about where you are in space. It is the silent sense, the one you only notice when it fails. Having now grasped the physiology of hearing, we are perfectly prepared to appreciate its sister system. So when we return, we will shift our focus from the snail shell of the cochlea to the loops and chambers of the vestibule. Our next lesson will hand us the adjacent piece, which is equilibrium and balance, the vestibular system. Lesson 6, Equilibrium and Balance, the vestibular system. We have spent our recent sessions exploring the vibrant world of light and the rhythmic world of sound, and in doing so, we have seen how the eye and the ear act as our primary windows to the external environment. But now, we turn our attention inward to a sense that is so fundamental, yet so silent, that we often forget it exists until it is momentarily disrupted. We are moving from the snail shell of the cochlea to its neighboring chambers within the inner ear's bony labyrinth. This lesson is an exploration of the vestibular system, your body's internal compass and level. This is the next natural step in our learning journey together. While hearing allows you to perceive the vibrations of the air, the vestibular system allows you to perceive yourself in relation to the world's most constant force, gravity. It is the system that tells you, even with your eyes closed and in total silence, whether you are standing upright, lying down, or leaning to the side. It is the silent guardian of your posture and the master of your stability. As we begin this lesson, I invite you to settle into your chair or bed and feel the weight of your body against the surface beneath you. That sensation of weight and position is the conversation we are about to study. Take a moment to let that idea find its place as we begin our breakdown of the anatomy and physiology of balance. To understand the vestibular system, we must look at the remaining sections of the inner ear that we bypassed during our study of hearing. Located just behind the cochlea, as within the same protective temporal bone, is the vestibular apparatus. This complex structure is divided into two main functional parts: the vestibule, which handles our sense of position and linear movement, and the semicircular canals, which handle our sense of rotation. Let us look first at the vestibule. This central egg-shaped cavity contains two membranous sacs called the utricle and the saccule. Inside each of these sacs is a small, specialized patch of sensory tissue known as a macula. Think of the macula as a microscopic level, like those used by a carpenter. It consists of a layer of hair cells, whose tiny stereocilia are embedded in a thick, jelly-like substance called the otolithic membrane. But here is the fascinating detail. Sitting on top of this jelly are thousands of tiny calcium carbonate crystals called otoliths, or ear stones. The logic of this system is purely mechanical and beautifully simple. When you tilt your head or move forward in a car, the weight of these tiny stones causes the jelly-like membrane to slide in the direction of the movement. This sliding motion bends the stereocilia of the hair cells underneath. As we learned in our study of hearing, when these hairs bend, they open ion channels that create an electrical signal. This is how your brain knows which way your head is tilted relative to the ground. The utricle is oriented horizontally, so it detects forward and backward movements, while the saccule is oriented vertically, detecting up and down movements, like the sensation of an elevator starting or stopping. Now contrast this with the semicircular canals. These are three bony loops, each oriented in a different plane of space. One for nodding yes, one for shaking your head no, and one for tilting your head toward your shoulder. At the base of each loop is a swollen area called the ampulla. Inside the ampulla sits a structure called the crista ampullaris. Unlike the macula, the crista does not have stones. Instead, it features a tall gelatinous cap called the cupula that stretches from the hair cells all the way to the roof of the ampulla. The semicircular canals are filled with a fluid called endolymph. When you rotate your head, the fluid inside the corresponding canal doesn't move immediately because of inertia. It lags behind. This lagging fluid pushes against the tall, sail-like cupula, causing it to bend. This bending, in turn, stimulates the hair cells at its base. If you have ever spun in a circle and then stopped suddenly, the fluid kept moving for a few seconds, pushing the cupula in the opposite direction and making you feel like you were still spinning. This is the mechanism of dynamic equilibrium. It is a sophisticated way of measuring the velocity and direction of your head's rotation in three-dimensional space. If your mind is still following along, try visualizing this. Your vestibule is the steady level that monitors the constant pull of the earth, while your semicircular canals are the gyroscopes that monitor every turn, twist, and tilt. Together, they provide a continuous high-speed data stream to your brain, ensuring that you remain upright and oriented no matter how the world moves around you. This is a good place to rest your thoughts for a moment before we continue. Again, if we dive deeper into the neural side of this system, we find that the information from the hair cells in the maculae and cristae travels along the vestibular nerve, which joins the cochlear nerve to form the vestibulo-cochlear nerve, cranial nerve 8. This nerve delivers the data to two main processing centers in the brain: the vestibular nuclei in the brainstem and the cerebellum. The cerebellum is the brain's master coordinator of motor movement. It doesn't just receive vestibular data, it compares it with information coming from your eyes and from the proprioceptors in your muscles and joints. If you are standing on a moving bus, Your cerebellum is the reason you don't fall over. It takes the unstable signals from your inner ear and your feet and calculates the exact muscle contractions needed to keep you balanced. It is a real-time, subconscious physics engine that runs in the background of your life every single second. One of the most remarkable outputs of this system is something called the vestibulo-ocular reflex, or VOR. Have you ever noticed that you can keep your eyes perfectly fixed on a single word on a page, even while you are shaking your head back and forth? This is the VOR in action. When your vestibular system detects your head moving to the left, it sends an immediate automatic signal to the muscles of your eyes to move them to the right at the exact same speed. This counter-rotation of the eyes keeps the image perfectly steady on your retina. Without this reflex, the world would look like a shaky, blurred home movie every time you took a step. Historically, our understanding of this system was a slow discovery. Early anatomists thought the semicircular canals were part of the hearing mechanism, perhaps acting as a sort of echo chamber. It wasn't until the 19th century that researchers like Floerens and Purkinje began to realize that damaging these canals led not to deafness, but to a total loss of coordination and a terrifying sense of vertigo. They began to see that the inner ear was a dual-purpose organ, housing two very different senses in the same small space. Take a moment to let that idea find its place. The same fluid, endolymph, that carries the vibrations of a symphony also carries the information of your physical orientation. They share the same pipes and the same electrical cables. But your brain is so sophisticated that it never confuses the two. It is an extraordinary example of biological multitasking. If your mind is wandering, try visualizing those tiny otoliths, those ear stones, resting quietly in the jelly of your utricle right now. As you lie still, they are pulled straight down by gravity, sending a steady, reassuring signal to your brain that you are safely at rest. So you see, this isn't just an abstract map of a hidden organ. It is the principle that explains why we experience motion sickness. Motion sickness is essentially a disagreement between your special senses. Imagine you are in the cabin of a ship during a storm. Your vestibular system, specifically those semicircular canals and maculae, feels every roll and pitch of the waves. It is screaming to your brain that you are moving violently. However, if your eyes are fixed on the stationary walls of the cabin, they are sending a signal that you are perfectly still. Your brain receives these two conflicting messages and, for reasons scientists are still debating, interprets this sensory mismatch as a sign of poisoning, leading to nausea. This is why looking at the horizon, giving your eyes a moving reference point that matches what your ears feel, can often help clear the feeling. This system also explains the sensation of vertigo. In many cases, vertigo is caused by a condition called benign paroxysmal positional vertigo, or BPPV. This happens when one of those tiny ear stones, or otoliths, becomes dislodged and drifts into one of the semicircular canals where it doesn't belong. When you move your head, that loose stone rolls through the fluid and hits the cupula, telling your brain you are spinning even when you aren't. It is a physical error in the body's internal gyroscope. Furthermore, we can see the vital importance of the vestibular system in our aging population. As we get older, the hair cells in the vestibule can decrease in number, and the otoliths may become less stable. This is why falls are such a significant health concern for the elderly. Their internal level is not as sharp as it once was, and the cerebellum has a harder time calculating those quick, corrective movements. By understanding this, we can design better balance exercises and physical therapy to help keep people steady on their feet. Finally, consider the challenge faced by astronauts in zero-G. In space, there is no gravity to pull the otoliths down. The level in the utricle and saccule becomes useless, floating in the jelly without a down to point to. Astronauts must rely entirely on their eyes and their semicircular canals for orientation, which is why space adaptation syndrome, or space sickness, is so common in the first few days of a mission. Their brains have to learn a completely new set of rules for what it means to be balanced. As you rest tonight, appreciate the quiet pull of the earth. It is the anchor for your vestibular system, the steady north for your internal compass. We have now explored the mechanics of sight, the vibrations of sound, and the steady gravity of balance. You have seen how the inner ear serves two masters, translating both the air's movement and the body's motion into a language the brain can comprehend. We have completed our study of the mechanical and wave-based senses, the physics of the human body. But there is another world of sensation that is perhaps even more intimate and more ancient than sight or sound. It is a world not of waves or gravity, but of molecules and chemistry. Having now grasped the physiology of balance, we are perfectly prepared to transition from the world of physics to the world of chemistry. Our next lesson will lead us away from the ears and toward the nose, where we will explore how floating molecules become the scents that trigger our deepest memories. So, when we return, we will begin our study of olfaction, the sense of smell. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the gateway to the world of chemicals. Lesson 7. Olfaction, the sense of smell. In our previous session, we explored the inner ear's silent guardian, the vestibular system. We watched as gravity and motion were translated into a sense of balance, providing us with a steady anchor in a moving world. But now, we move away from the world of physics, away from waves, pressures, and the pull of the earth, and we step into the ancient, intimate world of chemistry. We are beginning our study of olfaction, the sense of smell. This is the next natural step in our learning journey because it introduces us to our first chemical sense. While sight and hearing are distal senses that allow us to perceive things from a great distance, smell is a bridge that brings the molecular reality of the world directly into our bodies. It is perhaps our most primal sense, deeply rooted in the parts of our brain that governed the survival of our ancestors long before we developed complex language or abstract thought. In this lesson, we will explore how floating molecules in the air are captured by a small hidden patch of tissue in your nose and transformed into scents that can make your mouth water, your heart race, or your mind drift back to a forgotten childhood memory. Take a moment to let that idea find its place. You are about to discover how your nose reads the invisible chemical book of the world. To understand olfaction, we must first look at the laboratory where the chemicals are sampled. This happens high in the roof of your nasal cavity, in a region called the olfactory epithelium. Imagine this as a small yellowish-brown patch of tissue, roughly the size of a postage stamp, tucked away in a place where the air you breathe doesn't even travel directly. To get a good sample of a scent, you often have to sniff deeply, drawing the air upward into this hidden alcove. The olfactory epithelium is a bustling community made of three primary types of cells. First, there are the olfactory sensory neurons. These are the stars of the show, the actual chemoreceptors. What makes them unique is that they are among the few neurons in the entire human body that regularly die and are replaced throughout your life. Surrounding them are supporting cells, which provide physical structure and nourishment, much like the glial cells in your brain. Finally, there are basal cells, which act as the nursery, stem cells that divide to create new sensory neurons when the old ones wear out. For you to smell something, the substance must be volatile, meaning it must be in a gaseous state so it can float through the air and enter your nose. But that is only the first step. Once the molecule reaches that hidden stamp of tissue, it must dissolve into the layer of mucus that coats the epithelium. Think of this mucus not as a nuisance, but as the essential solvent of memory. If the molecule cannot dissolve, the receptors cannot see it. On the surface of the sensory neurons are tiny, hair-like projections called olfactory cilia. These cilia are covered in the specialized receptor proteins. When a dissolved odorant molecule finds a receptor that matches its specific shape, they bind together like a key fitting into a lock, or perhaps more accurately, like a specific puzzle piece finding its home. This binding event triggers the process of transduction. Inside the cell, the binding of the odorant to the receptor sets off a relay race. It activates a protein called a G protein, which then stimulates an enzyme to produce a messenger molecule called cyclic AMP, or CAMP. This messenger then travels to the cell's membrane and opens up ion channels, allowing sodium and calcium ions to rush in. This causes the neuron to depolarize, creating an electrical signal. Don't worry about memorizing the chemical names. Just let the logic of it wash over you. A floating molecule touches a hair, a protein changes shape, a messenger is sent, and suddenly the chemical has become electrical. The invisible landscape has been translated into a message for the brain. Now that the signal has been created, it must be organized. The axons of these thousands of sensory neurons travel upward through tiny holes in the cribriform plate, a thin, sieve-like bone at the base of your skull. On the other side of this bone lies the olfactory bulb, an extension of the brain itself. Here, the raw data is sorted in a way that is truly masterful. Inside the olfactory bulb are spherical structures called glomeruli. You can think of a glomerulus as a sorting office. All the neurons that respond to a specific type of odorant, say the molecule that makes a rose smell like a rose, converge on the same specific glomerulus. This creates a smell map in the bulb. A complex scent, like the smell of a forest after rain, isn't just one signal. It is a specific chord played across many different glomeruli at once. Now, your brain recognizes the pattern of which notes are being played and identifies the scent accordingly. From the glomeruli, the signal is passed to mitral cells, which act as the final processing unit before the message leaves for the deeper parts of the brain. But here is where olfaction breaks all the rules of the nervous system. As we have learned in almost every other lesson, sensory information usually goes to the thalamus, first to be filtered and sorted. But the sense of smell has a direct hotline connection to the olfactory cortex and, more importantly, to the limbic system. The limbic system is the emotional center of your brain. It includes the amygdala, which processes fear and excitement, and the hippocampus, which is the seat of long-term memory. This direct anatomical link is why a scent can bypass your logic entirely. You might smell a certain type of old paper and be instantly flooded with the feeling of your grandmother's attic, or smell a specific perfume and feel a sudden jolt of nostalgia or even sadness, long before your conscious mind identifies what the smell actually is. If your mind is still following along, try visualizing this. Your nose isn't just telling you what is in the air, it is whispering directly to your heart and your history. Take a moment to let that idea find its place. This direct connection is a relic of our evolutionary past. For an early human, the smell of a predator, the scent of a mate, or the aroma of a poisonous plant needed to be processed instantly without the delay of thinking about it. Survival depended on an immediate, emotional reaction to the chemical world. Even today, though we live in a world of screens and steel, our brains still treat the sense of smell with that same ancient high-priority respect. So you see, this isn't just a lesson in anatomy. It is the principle that explains why flavor is so much more than just taste. When you eat a strawberry, your tongue only detects the sweetness and the acidity. The strawberryness, the complex floral and fruity essence, comes from the molecules traveling from the back of your mouth up into your nasal cavity. This is called retronasal olfaction. This is why when you have a cold and your olfactory epithelium is buried under a layer of thick mucus, your food tastes like cardboard. The chemistry of the meal is still hitting your tongue, but the poetry of the meal, the smell, is being blocked. This knowledge also explains the power of aromatherapy and the use of scents for relaxation. Because smell has a direct line to the emotional brain, certain molecules, like those found in lavender or sandalwood, can physically lower your heart rate and reduce cortisol levels. They aren't just pleasant smells. They are chemical keys that unlock specific physiological responses in your nervous system. As you prepare for sleep tonight, the subtle scents in your room are actively participating in the way your brain winds down. We must also consider the condition known as anosmia, the loss of the sense of smell. This can happen due to head trauma, which can shear the delicate axons as they pass through the cribriform plate, or due to viral infections that damage the sensory neurons. For those living with anosmia, the world becomes a flatter place. The emotional depth of life, the warning sign of smoke, and the joy of a shared meal are all diminished. It reminds us of the profound value of this often overlooked sense. Finally, think about the social chemistry of our lives. While humans don't rely on pheromones as much as other animals do, we are still subtly influenced by the chemical signals given off by those around us. We are constantly, subconsciously reading each other through scent. It is a reminder that we are biological beings, deeply embedded in a chemical environment. By understanding olfaction, we gain a deeper appreciation for the invisible layers of reality that surround us every second of every day. Let's hold that thought here, a complete piece of the puzzle. You have now explored the hidden laboratory of the nose. You have seen how molecules are captured in a solvent of memory, translated through a G-protein relay, and sent on a direct hotline to the emotional centers of your brain. You understand that smell is the sense of patterns, chords, and deep ancient connections. But as we have touched upon, smell is only half of the story of how we sample the chemistry of our world. Smell and taste are like two sides of a single coin, working in such close harmony that we often can't tell where one ends and the other begins. Having now grasped the physiology of olfaction, we are perfectly prepared to descend from the roof of the nasal cavity down to the surface of the tongue. Our next lesson will hand us the adjacent piece as we explore the world of sweetness, saltiness, and the savory depths of umami. So, when we return, we will begin our study of gustation, the sense of taste. This is a good place to rest your thoughts for a moment before we continue our journey into the flavors of life. Lesson 8. Gustation, the sense of taste. In our last session, we climbed into the hidden attic of the nasal cavity and explored the ethereal world of olfaction. We saw how a single floating molecule can trigger a cascade of memories and emotions, acting as a direct hotline to the heart of the brain. But now, we descend from the roof of the nose to the floor of the oral cavity. We are moving from the sense of smell to its inseparable companion, gustation, or the sense of taste. While smell allows us to sample the world from a distance, taste is the ultimate, intimate gatekeeper. It is the final check, the last biological yes or no, before we allow the outside world to truly become part of our inside world through the act of digestion. This is the next natural step in our learning journey because taste and smell are the twin pillars of our chemical perception. In this lesson, we will look at the landscape of the tongue, the microscopic anatomy of the taste bud, and the 5 primary languages of flavor that our body speaks. We aren't just looking at why a strawberry is sweet. We are looking at the sophisticated survival mechanism that identifies energy, monitors minerals, and guards against poison. Take a moment to let that idea find its place. Your tongue is a sentinel, a highly trained chemist standing watch over your health and your pleasure. As you rest tonight, imagine the surface of your tongue as a vast, sensitive terrain, waiting to decipher the secrets of the next thing you consume. To understand gustation, we must first look at the landscape of the tongue. When you look in a mirror and see those tiny bumps on your tongue, you might think those are the taste buds themselves, but that is a common misconception. Those bumps are actually called papillae. Think of papillae as the mountains and valleys of the tongue, and the taste buds are the hidden residents living within those valleys. There are 3 main types of papillae that house our sense of taste. The fungi-form papillae are shaped like tiny mushrooms and are scattered across the entire surface of the tongue. The vellate papillae are much larger and form a V shape at the very back of the throat. Finally, there are the foliate papillae, which sit in small trenches on the sides of your tongue. Nested within the walls of these papillae are the taste buds. Each taste bud is an oval-shaped cluster of about 50 to 100 specialized cells. You might picture a taste bud like a tiny orange, with its cells arranged like segments. There are 3 types of cells here. The gustatory cells, which are the actual receptors. The supporting cells, which provide structure. And the basal cells, which are the stem cells. Just like the neurons in your nose, these gustatory cells are constantly being replaced every 10 days or so, worn down by the heat and friction of the food we eat. At the very top of each taste bud is a tiny opening called the taste pore. From this pore, long microvilli, we call them gustatory hairs, extend out into the saliva. For you to taste anything, the substance must be a saporant, meaning a chemical that can dissolve in water. This is why your saliva is so essential. So if your tongue were perfectly dry, you could place a sugar cube on it and taste absolutely nothing. The saliva acts as the medium, dissolving the chemicals so they can flow into the taste pore and touch the gustatory hairs. Once a molecule binds to a receptor on a gustatory hair, the process of transduction begins, but it happens in two very different ways, depending on what you are tasting. If your mind is still following along, try visualizing the two different languages of taste transduction. The first is the ion language, used for salty and sour tastes. Salty taste is the simplest. It is triggered by sodium ions. When you eat salt, the sodium flows directly through open channels into the gustatory cell. Sour taste is triggered by hydrogen ions from acids. These ions also flow directly into the cell or block potassium channels. In both cases, the influx of positive charge causes the cell to depolarize, releasing a neurotransmitter that tells your brain this is salty or this is sour. The second language is the G protein language, used for sweet, bitter, and umami. These molecules are too large or complex to flow through channels. Instead, they bind to receptors on the outside of the hair, which then activates an internal messenger called Gustation. This messenger sets off a chemical chain reaction inside the cell, eventually leading to the release of neurotransmitters. Whether it is an ion or a G protein, the result is the same. The chemical identity of your food is converted into an electrical signal that races toward your brain. If we dive deeper into the history and nuances of these tastes, we find that for a long time, Western science only recognized 4 primary tastes: sweet, sour, salty, and bitter. It wasn't until the early 20th century that a Japanese chemist named Kikunai Ikeda identified a 5th distinct taste. He was eating a bowl of dashi, a traditional kelp broth, and realized its savory, meaty quality didn't fit into the other 4 categories. He identified the chemical responsible as L-glutamate and named the taste umami, which roughly translates to deliciousness or savory. It took nearly a century for the rest of the scientific world to officially recognize umami, but we now know it has its own dedicated receptors. It is the taste that tells your body you are consuming protein and amino acids, the building blocks of life. Another layer to peel back is the persistent myth of the tongue map. You may have seen diagrams in school suggesting that you only taste sweet on the tip of your tongue or bitter at the very back. We now know this is entirely incorrect. While some areas of the tongue may have slightly lower thresholds for certain tastes, every part of the tongue that has taste buds can detect all 5 tastes. The brain doesn't look at where on the tongue the signal came from to determine the flavor. It looks at which specific nerve fiber is firing. This brings us to the gustatory pathway. The signals from your tongue don't travel along just one nerve. The front, 2/3 of the tongue, are served by the facial nerve, cranial nerve 7. The back third is served by the glossopharyngeal nerve, cranial nerve 9. And for the receptors in the very back of the throat and the epiglottis, we use the vagus nerve, cranial nerve 10. This triple redundancy ensures that even if one pathway is damaged, your ability to taste remains largely intact. These nerves carry the signal to the medulla oblongata, specifically to a region called the solitary nucleus. This is a good place to rest your thoughts for a moment before we continue. Think of the solitary nucleus as the first sorting station. From here, some signals trigger immediate reflexes like salivation or, in the case of something very bitter, a gag reflex. Other signals continue upward to the thalamus, the master relay station, and finally arrive at the primary gustatory cortex in the insula of the brain. It is here that the signal becomes a conscious experience. You don't just detect sweetness, you perceive it, and perhaps you even associate it with a specific memory or desire. Don't worry about memorizing the numbers of the cranial nerves. Just let the logic of the journey wash over you. Your tongue is sending a constant multi-channeled report to your brain, not just about what is in your mouth, but about the chemical safety and nutritional value of your environment. It is a dialogue that has been happening since the very first day you were born, and it is one of the most consistent ways your body interacts with the physical world. So you see, this isn't just an exercise in biological labeling. It is the principle that explains why we have such a sweet tooth and such a natural aversion to bitter. From an evolutionary perspective, sweetness is the signal for carbohydrates and high-energy calories, things that were rare and precious for our ancestors. Bitter, on the other hand, is the taste of most natural toxins and alkaloids found in poisonous plants. Our intense dislike of bitter things as children is actually a survival mechanism. Our tongues are trying to protect us from accidental poisoning until we learn which bitter things, like coffee or dark greens, are actually safe. This knowledge also explains why flavor is such a multi-sensory experience. As we have discussed, about 80% of what we call flavor is actually smell. But it is even more complex than that. Your brain also takes into account the texture of the food, which is sensed by mechanoreceptors, and the temperature, which is sensed by thermoreceptors. And this information is carried by the trigeminal nerve, the same nerve that senses touch and pain in your face. This is why a hot chili pepper feels like it is physically burning you. The chemical capsaicin actually triggers the same pain receptors that respond to literal fire. Flavor, then, is a grand synthesis of chemistry, texture, temperature, and aroma. We can also see the clinical relevance of gustation in the way our taste changes as we age. As we get older, our basal cells divide less frequently, and the number of active taste buds decreases. This is why many elderly people find their food to be bland and may begin to oversalt their meals. Furthermore, certain medications or a deficiency in zinc can lead to hypogeusia, a reduced ability to taste. Because taste is so linked to our appetite and our enjoyment of life, a loss of taste can lead to significant weight loss and depression. Finally, think of the way the modern food industry uses this physiology. By understanding exactly how the salty and sweet receptors work, scientists can create bliss points, combinations of sugar, salt, and fat that maximize the dopamine response in the brain. By understanding the logic of the tongue, we can see how we are sometimes manipulated by our own biology. As you rest tonight, appreciate the simple, honest work your tongue does. It is a faithful servant, guiding you toward what nourishes you and away from what might harm you, one molecule at a time. Let's hold that thought here. A complete piece of the puzzle. You have now journeyed from the roof of the nose to the floor of the mouth. You have seen how ions flow and G proteins trigger, and you have followed the signal through the cranial nerves to the gustatory cortex. You understand that taste is the final intimate gatekeeper of your biology. But our study of the special senses is not yet complete. We have looked at these systems as individual islands of information, the eyes, the ears, the nose, and the tongue. But you don't live in a world of isolated sounds or lonely colors. You live in a unified reality where the smell of the coffee matches the heat of the cup and the darkness of the liquid. Having now grasped the physiology of each individual sense, we are perfectly prepared to see how the brain brings them all together. Our next lesson will hand us the adjacent piece as we explore the grand conductor of the nervous system. So, when we return, we will begin our study of the integration of the special senses in the central nervous system. This is a good place to rest your thoughts for a moment before we see how the pieces of the puzzle form a single, beautiful picture. Lesson 9. Integration of the special senses in the central nervous system. In our previous lessons, we have journeyed through the individual islands of the human experience. We have explored the vibrant light capture of the eye, the rhythmic mechanical dance of the ear, the gravity-defying logic of the vestibule, and the chemical signatures found in the nose and on the tongue. But as you rest now, I want you to realize that you do not live in a world of isolated sensations. You do not experience just a color or just a sound. Instead, you live in a rich, unified tapestry of reality. In this 9th lesson, we are going to look at the grand conductor of this symphony, the central nervous system. This is the next natural step in our learning journey because it is here, within the folds of the brain, that the separate streams of data are woven together into a single, coherent story. We are moving from the sensors to the processor. We will look at how the brain organizes this massive influx of information, how it filters out the noise of the world, and how it creates the three-dimensional, multi-sensory now that you are experiencing even as you drift toward sleep. Take a moment to let that idea find its place. Your brain is not just receiving the world, it is actively constructing it for you. If your mind is still following along, try visualizing those 5 separate rivers of information finally meeting in a single, deep ocean of consciousness. To begin our breakdown, we must revisit a structure we have mentioned many times, the thalamus. Located deep in the center of the brain, the thalamus is often called the gateway to the cortex. Except for the sense of smell, which has its own ancient direct root, every single piece of special sensory information must pass through the thalamus before it can reach your conscious mind. Think of the thalamus as a master switchboard operator or a highly discerning editor. It receives thousands of electrical signals every millisecond, and its job is to decide which ones are important enough to be sent upstairs to the cerebral cortex. When a signal arrives at the thalamus, it is sent to a specific relay nucleus dedicated to that sense. For instance, visual signals from the optic nerve go to the lateral geniculate nucleus, while auditory signals from the ear go to the medial geniculate nucleus. From these specific points, the information is dispatched to the primary sensory cortecias. The primary visual cortex in the occipital lobe receives the raw data of edges, colors, and motion. The primary auditory cortex in the temporal lobe receives the frequencies and rhythms of sound. The primary gustatory cortex in the insula receives the chemical signals of taste. But if the process stopped there, the world would be a confusing jumble. You would see a yellow shape and hear a crunching sound, but you wouldn't know you were eating an apple. This is where the sensory association areas come into play. Surrounding each primary cortex is a secondary area that interprets the raw data. The visual association area, for example, compares the yellow shape you are seeing to your memories of fruit, while the auditory association area identifies the crunching sound as the specific texture of a gala apple. The final and most complex step of this integration happens in the multimodal association areas, such as the posterior parietal cortex. This is where the brain performs the magic of binding. It takes the yellow from the occipital lobe, the crunch from the temporal lobe, and the sweetness from the insula, and it merges them into a single concept. I am eating a delicious apple. This is a good place to rest your thoughts for a moment before we continue. Don't worry about memorizing the names of every lobe. Just let the logic of the merging wash over you. Your brain is a master weaver, constantly stitching together the threads of your senses into a single, beautiful cloth. If we dive deeper into this integration, we encounter one of the most fascinating challenges in neuroscience, known as the binding problem. Scientists still wonder exactly how the brain keeps all these signals synchronized. When you watch a person speak, the light from their lips reaches your eyes much faster than the sound of their voice reaches your ears. Furthermore, the neural processing for vision is slightly slower than the processing for hearing. Yet, in your conscious experience, the lips and the voice are perfectly in sync. Your brain actually buffers this information, holding on to the faster signal for a few milliseconds so that it can present them to your consciousness at the exact same time. It is like a sophisticated video editing suite that ensures the audio and visual tracks are perfectly aligned. We must also consider the role of the midbrain, specifically the superior and inferior colliculi. These structures handle our unconscious sensory integration. The superior colliculus receives input from both the eyes and the ears. Its job is to coordinate space. If you hear a sudden loud noise to your right, the superior colliculus automatically triggers your neck muscles to turn your head and your eye muscles to look in that direction. It creates a spatial map where sounds and sights are pinned to the same coordinates. This is why you don't have to think about where a sound came from. Your body has already oriented itself toward the source before you are even fully aware of what happened. Historically, our understanding of this integration was shaped by the study of synesthesia. This is a rare condition where the wires of the sensory integration areas are more interconnected than usual. A person with synesthesia might see a specific color every time they hear a certain musical note, or taste a specific flavor when they read certain words. By studying these individuals, researchers realize that our brains are naturally designed to link senses together. It's just that for most of us, those links happen behind the scenes, below the level of conscious awareness. Take a moment to let that idea find its place. Even for those of us without synesthesia, our senses are constantly talking to each other. If you see a picture of a lemon, your mouth might start to water. That is your visual cortex communicating directly with your gustatory and salivary centers. If your mind is still following along, you try visualizing these neural pathways as busy highways, with messages constantly crossing over from one lane to another, enriching your experience of the world. So you see, this integration is the principle that explains why our experiences are so much more than the sum of their parts. A classic example of this is the McGurk effect. This is a famous psychological phenomenon where what you see someone's mouth doing can actually override what you hear them saying. If a video shows a person saying the sound effa, but the audio is actually the sound bia, your brain will often resolve the conflict by making you hear fa. Your brain trusts its visual integration so much that it will actually rewrite the auditory data to make sense of the scene. This isn't just a laboratory trick. It's what allows you to understand a friend in a noisy restaurant by watching their lips. This integration also explains the profound concept of flavor. As we have discussed in earlier lessons, what we call flavor is not just taste. It is a grand synthesis of gustation from the tongue, olfaction from the nose, and somatosensation, the texture and temperature from the mouth. But it goes even further. The crunch of a potato chip, an auditory signal, is an essential part of the flavor experience. If you were to eat a stale chip that tasted the same but made no sound, your brain would tell you the flavor is wrong. The integration of sound and taste is what tells your brain the food is fresh and safe to eat. Furthermore, we see this synthesis in how we navigate our physical environment. To walk down a flight of stairs, and your brain must integrate the visual map of the steps, the vestibular level of your balance, and the proprioceptive feeling of your feet touching the ground. If any one of these signals is missing or mismatched, like when you think there is one more step than there actually is, the integration fails, and you experience that sudden, jarring jolt of disorientation. It is a reminder of how much we rely on the grand conductor to keep our world steady and predictable. As you prepare for sleep, consider how your brain is currently integrating the sensations of your environment. It is taking the low hum of a fan, the coolness of the air on your skin, the weight of your blankets, and the soft rhythm of your breathing, and it is weaving them into a blanket of safety. It is filtering out the ticking of a clock or the distant sound of traffic so that you can focus on the internal sensation of relaxation. This is the ultimate gift of sensory integration. It allows you to feel at home in your own body. Let's hold that thought here, a complete piece of the puzzle. You have now seen how the individual signals from our special senses are gathered by the thalamus, dispatched to the primary lobes, and finally merged in the association areas of the brain. You understand that your reality is a carefully constructed masterpiece, a unified story told by the central nervous system. We have seen the system in its magnificent, integrated perfection. But as with any complex machine, there are times when the parts or the connections may face challenges. To truly appreciate the wonder of the special senses, we must also understand what happens when the light grows dim, the sound fades, or the balance falters. Our final lesson will hand us the last piece of the puzzle as we explore the clinical side of our study. So, when we return, we will look at how we diagnose, treat, and adapt to the various conditions that can affect these delicate systems. We will conclude our journey by looking at disorders of the special senses and clinical correlations. This is a good place to rest your thoughts for a moment before we complete our study together. Lesson 10. Disorders of the Special Senses and Clinical Correlations. We have traveled a long road together, my friend. From the very first spark of light hitting a photoreceptor to the complex chemical dance on the surface of your tongue, we have explored the magnificent machinery that allows you to experience the world. But as we reach this 10th and final lesson, we must look at the special senses through a different lens, one of care, resilience, and clinical understanding. In this lesson, we are going to explore what happens when these delicate systems face challenges. We will look at the disorders that can dim the lights, muffle the music, or tilt the steady ground beneath our feet. This is the next natural step in our learning journey because understanding the pathology of a system is often the best way to truly appreciate its healthy perfection. We are moving from the how it works to the what happens when it struggles. We will look at common conditions like glaucoma, cataracts, and hearing loss, not as cold medical diagnoses, but as stories of biological wear and tear, and often as stories of incredible human adaptation. Take a moment to let that idea find its place. As we conclude our study, I want you to feel a sense of empowerment. Knowledge of these conditions is the first step in protecting the precious windows through which you view your life. If your mind is still following along, let us take this final walk through the clinical landscape of the senses. To begin our breakdown of sensory disorders, we must return to the eye, which is perhaps the most vulnerable of our special sense organs. Most vision problems are not actually diseases in the traditional sense, but rather physics errors, what we call refractive errors. Think of the eye as a finely tuned optical instrument. If the eyeball is just a fraction of a millimeter too long, the light focuses in front of the retina instead of on it. We call this myopia, or nearsightedness. If the eyeball is too short, the light reaches the retina before it has finished focusing, leading to hyperopia, or farsightedness. Then there is astigmatism, where the cornea is shaped more like a football than a basketball, causing light to blur in multiple directions. These aren't failures of biology so much as subtle deviations in geometry. Moving deeper into the eye, we encounter conditions that affect the clarity of our vision. A cataract is a clouding of the lens. You might picture the lens of your eye as a clear pool of water. Over time, the proteins within that water can begin to clump together, making the water murky. This happens slowly, often over decades, and it feels like looking through a frosted window or a piece of waxed paper. On the other hand, glaucoma is a much more silent and serious threat. It is caused by an increase in intraocular pressure. As we learned, the aqueous humor must drain constantly to maintain the right pressure. If the drainage pipe, the scleral venous sinus, becomes blocked, the pressure builds up and begins to physically crush the delicate fibers of the optic nerve. Because this usually starts at the edges, a person can lose a great deal of their peripheral vision before they even realize something is wrong. When we turn to the ear, we divide disorders into two main categories: conduction deafness and sensor neural deafness. Conduction deafness is a mechanical problem. It occurs when something prevents sound vibrations from reaching the inner ear. This could be a simple buildup of earwax, a torn eardrum, or a condition called otosclerosis, where the tiny ossicles, the hammer, anvil, and stirrup, become fused together and can no longer vibrate. Sensor neural deafness, however, is a neural problem. It happens when there is damage to the hair cells in the cochlea or to the auditory nerve itself. This is often the result of long-term exposure to loud noise or the natural aging process. Finally, we must look at the chemical senses. Anosmia is the loss of the sense of smell. This can be temporary, such as when a cold causes inflammation in the nasal cavity, or permanent, such as when a head injury shears the delicate olfactory nerves as they pass through the skull. Because smell and taste are so closely linked, people with anosmia often complain that they have also lost their sense of taste, though what they have really lost is the flavor or the aroma of their food. True loss of taste, or ageusia, is much rarer, as it would require damage to 3 different cranial nerves simultaneously. If we dive deeper into the clinical world, we find that many of these disorders are deeply tied to the narrative of aging. We have a specific term for the age-related loss of near vision, presbyopia. As we get older, the lens of the eye becomes less elastic, and the ciliary muscles struggle to bulge it for close-up work. This is why many people find themselves holding a book at arm's length as they enter their 40s or 50s. It is a natural, almost universal part of the human journey. Similarly, presbycusis is the gradual loss of high-frequency hearing that comes with age. It often begins so subtly that the person doesn't notice it, but they may find it increasingly difficult to understand conversation in a crowded room or to hear the high-pitched voices of children. Historically, the treatment of these conditions has seen a dramatic evolution. In ancient times, cataract surgery involved a technique called couching, where a needle was used to simply push the clouded lens out of the way and into the back of the eye. While it allowed light back in, the vision remained incredibly blurry. Today, we replace the clouded lens with a high-tech synthetic one in a procedure that takes mere minutes. This is a testament to how far our understanding of ocular anatomy has come. We have moved from simply moving the obstacle to restoring the function. One of the most heartbreaking conditions we study in the deeper layers is macular degeneration. The macula is the very center of your retina, the part responsible for your sharpest, most detailed vision. In dry macular degeneration, the cells of the macula slowly break down, often due to the accumulation of metabolic waste products called drusen. In the wet form, abnormal blood vessels grow underneath the retina and leak fluid. Unlike glaucoma, which takes away the edges of your world, macular degeneration takes away the center. It makes it impossible to recognize a face or read a line of text, even though the peripheral vision remains intact. It is a profound change in how a person navigates their environment. Take a moment to let that idea find its place. Whether it is the pressure of glaucoma or the clumping of proteins in a cataract, these disorders are often just extreme versions of normal biological processes. The body is always trying to maintain balance, but sometimes the repair mechanisms can't quite keep up with the wear and tear. If your mind is wandering, try visualizing the hair cells in your cochlea. They are like the grass in a well-trodden path. A little traffic, sound is fine, but a stampede, loud noise, can flatten them forever. This is why prevention and protection are the most important tools in our clinical arsenal. So you see, this final lesson is where the science of anatomy meets the reality of daily life. It is the principle that explains why we wear sunglasses to protect the proteins in our lenses from UV damage that could lead to cataracts. It explains why we wear earplugs at a concert, to prevent the mechanical overload that destroys hair cells. This knowledge isn't just for doctors. It's for everyone who wants to maintain their connection to the world. This synthesis also helps us understand the emotional and social impact of sensory loss. We often underestimate the sense of smell, but for someone with chronic anosmia, the world can feel flat and dangerous. They cannot smell smoke in the house or tell if food has gone bad. Similarly, the social withdrawal, often seen in the elderly, is frequently linked to untreated hearing loss. Yet if you cannot follow a conversation, the world becomes a lonely place. By identifying these clinical correlations, we can intervene with hearing aids, with surgery, or with simple environmental changes to keep people connected to their communities. Think of the way we have adapted our world to these clinical realities. We have developed Braille for those without sight, sign language for those without hearing, and sophisticated sensory integration therapies for children who find the world too loud or too bright. We are a resilient species. When one window closes, we find ways to open others. This is the ultimate lesson of the special senses. They are the tools we use to build our reality, but our reality is much bigger than any one tool. As you settle into the final moments of this course, consider your own sensory health. Perhaps you'll remember to blink more often when looking at a screen to keep your cornea moist. Or you'll be more mindful of the volume in your headphones. These small daily choices are the real-world application of everything we have studied. You are the steward of your own sensory temple. By understanding how it works and how it can struggle, you are better equipped to walk through the world with wonder and with care. We have reached the end of our curriculum on the special senses. You have journeyed from the anatomy of the eye to the physiology of the tongue, through the loops of the inner ear, and into the deep processing centers of the brain. We have seen the system in its health and in its challenges. You have gathered every piece of the puzzle, and you now hold a comprehensive map of how you perceive your existence. I want to thank you for your patience, your curiosity, and your presence. There is no more next lesson to prepare for tonight. Instead, I invite you to simply let all of this knowledge settle into the quiet corners of your mind. Don't worry about the terms or the diagrams now. Just hold on to the feeling of wonder for the body you inhabit. You are a biological masterpiece, and the world is waiting for you to experience it with fresh eyes and open ears when you wake. Rest deeply now, my friend. Your journey through the special senses is complete, and a well-earned sleep is the final lesson of the day.